I prepared 200 slides to give you a very in-depth science on our exosomes. And as usual, the topic is the quality, efficacy, and safety on our exosome technology. And I have a few lectures at IMCAS Paris later this month. So please join our lectures. I believe that Dr. Carlos and other great speakers will join our symposiums and lectures. And this is my topics for today, from number one quality to number nine clinical research. Uh, so last year I have been running more than 375,000 kilometers, and as Dr. Lee mentioned that ExoBio is still number one exosome company in the world because we have a little more increased number of scientific publications. Now we have 33, and we are expecting more than 5 publications during the rest of this year. And we have one more increase of our patent on the large stem cell exosomes. And so far, we have been generating about 2 million clinical cases worldwide for last 5 years. And this is last year award from Monaco. ExoBio had 5 awards, 3 product awards, and 2 doctors awards from Monaco. And as I mentioned, my team is generating more and more patents based on our scientific discovery. For example, very recently, we got one more patent on our rose stem cell exosomes as a cosmetic ingredient in European countries. This is the world first patent from any kind of plant exosomes. And I have one more update on our publication. You can see that the most well-known public journal on exosome is the Journal of Extracellular Vesicles, which has a impact factor 16. So it's a very high impact factor journal, and my team submitted one more manuscript on epidermic analysis contained in our exosomes. And I believe that you will see the publication in a few months. And we have one more publication accepted by the Journal of Cosmetic Dermatology, and the topic is on our large stem cell exosomes. Dr. Lydia from Poland, she submitted 8 cases. She showed the efficacy as well as safety of our large stem cell exosome products for a variety of clinical indications, actually. So I believe that in 2 months, you will see the actual publication. And I believe that Dr. Lydia will join IMCAS. So ExoBio is the only company committed to the quality production of exosomes from this ExoGMP facility. And we have been upgrading our process, and it's all about the quality and the safety. And from our scientific discoveries and from this quality control lab, we have all the instrument protocols to identify or to characterize the quality of our exosomes. So I hope all of you guys should be very cautious to choose the right product and the right technology and the right protocol for the quality control of your exosome product. You can see that our commitment to the quality of water and air. So based on these manufacturing process and our preparation, we believe that we are the best quality exosome product, but you can find a very high number of exosome copycats. We call that exosome mimicking or like products, but actually there are 5 types of exosome copycats you should be cautious about. So let me give you details one by one of these five categories. So what I'm showing here is that you should be very cautious to choose the right product, because many of other exosome claiming products are not based on pure exosomes, and their manufacturing is sometimes very dirty. So when you use that kind of product for your patient, you will see that this kind of very serious side effects. For example, here you see that from this publication, some doctors used uh, conditioned media from human stem cells, and they are not purified. They contain high impurities. You can see that when you use this kind of impure conditioned media, you can see very serious side effects. And here is another publication on the complications of the use of exosome copycats or very dirty exosome products. You will see that these kind of side effects when you use low quality or low priced, no publication exosome products. So I'm proposing these kind of items for you to think about to choose the right exosome technology from cell bank materials. Some companies are using cosmetic grade buyers or caps or other materials, but our product is a medical device grade, so it's the most or the safest quality of products, finished products. So please remember or screen capture this screen for you use to find the best quality product. I will go or I share all these information on our products in my lecture later. So in summary, you can see here that you can compare the left side, each our or ExcoBio manufacturing process. But on the right side, you can see that one action company who claimed they have GMP compliant process, which is not true. Clearly, you can see that our team is wearing the best quality of production grade costume or clothing. But on the right side, you can see that their process, their clothing, their instrument are research use grade or cosmetic grade. So you can see here the biggest differences from ExcoBio against other exosome competitors. And mostly, many exosome companies are based on conditioned media, as I mentioned earlier. Um, those kinds of conditioned media are containing cell debris, medium components, cellular waste, something like that. So you have to check the product registration or ingredient registration. You have to see the purified exosomes and their registration in the United States or in European countries, because conditioned media or that kind of impurities can cause serious inflammation. One important aspect or features you have to check is that you need to see this kind of on the website. This kind of clear lipid by membrane. The definition of exosome is or exosome should be surrounded by this kind of lipid by membrane, and we can share all these electron microscopic images to show clear lipid by membrane, as you can see here, and they can be delivered to human donor fibroblasts. But I will show you some different types of exosome claiming products. They don't show this kind of lipid by membrane image. And also, this is very scientific data, but let me tell you that you have to see these kind of exosome specific markers like CD63 or CD81 from their products. You can see that these kind of two peaks, two one black peak or one red peak, you can see. You have to see these kind of two different peaks from the exosome marker analysis. So our finished product can show these kind of exosome specific markers, but most exosome products you cannot see this kind of exosome specific markers. So here again, this kind of highly impure product or ingredient can cause inflammation. And also, let me show you another product which claims exosomes, but which is not actual exosome products. This product is based on conditioned media. They are not purified exosome. Even though conditioned media can contain very small fraction of exosomes, but this product is definitely based on conditioned media, or it's based on um, some kind of plant extract. You have to check the product registration. So this kind of products is already infringing some of our patents, and they are not actual exosomes, real exosomes, and they are not GMP compliant. They have no publication, they have no patents. So I hope all of you guys should check the product quality based on GMP manufacturing, publication, and patents, not only based on the price. This kind of product is a very cheap product because they have no R&D to produce or to generate a publication or any patent. And also sometimes you can see that on the market, synthetic exosomes. But when you look at the actual ingredients, they have no exosomes at all. You can see that collagen, HA, tree halose, and other peptides, something like that. They are not exosome products. They have no exosomes or extracellular vesicles. They have no registration. Scientifically, so far, their synthetic exosomes are not possible. And also, sometimes you can see that human or animal blood PRP exosome products, but they are not exosome products clearly because as you can see in the right side, when we look at this electron microscopic image, which is showing the blood, you can see that there are almost 2,000 lipoprotein or protein aggregates, but you have only two real exosomes because in the middle you can see that the size of lipoproteins like LDL, IDL, VLDL, or chylomicrons, they have almost the same size of exosome in the bottom, so they cannot be isolated from any kind of simple filtration process, and they are always co-isolated. If you want to isolate real exosome from any kind of blood, you should spend a lot of money, really, really big money. But it's all about diagnostics. But from any kind of simple filtration process, you are supposed to use only lipoproteins mostly. So you should be very cautious when you look at this kind of blood-derived or PRP exosomes. So here is another example which claims that they can isolate exosomes from the blood, but you can see that this device has only one 5 nanometer pore size filter, which cannot purify exosomes from the blood mixture. So if you use this kind of device, you are supposed to use very expensive lipoproteins or PRP. And sometimes you can see that kind of products based on milk exosomes. But when you look at this kind of publication which said that isolation and characterization of milk exosomes, they are showing that on the right side, you can see that this kind of table, you can see that they try to remove casein proteins or other impure proteins from the mixture of milk, but they can still cannot remove all the casein proteins because the size of casein protein is almost the same as the size of exosome. So you cannot remove all the casein proteins from milk. So when we look at this kind of panel in panel D, they are showing their electron microscopic image of the isolation or preparation of their exosomes. You cannot see any lipid by membrane. So it's very bad to say that milk exosomes or blood exosomes are pure, which is not pure ever. Also, when you look at this kind of ingredient list on the back of the product package, you cannot see any kind of exosome registration. There is no exosome registration, but you can see just colostrum, a type of milk. So you have to be very cautious. Also, when you look at fruits or vegetable extracts, you have to check the ingredient registration. These products are saying that they are exosomes, but when you look at the product ingredients of the package, you cannot see any exosomes. Also, when you look at liquid or frozen products, their long-term stability are not validated yet. We found one important data from a third-party company named Hansa Biomed. They compared lyophilized exosomes against liquid or frozen exosomes. So the key is that only lyophilized exosome stored at 4 degrees Celsius for 2 years, they maintained the original size of exosome, which is 100 nanometers. And other conditions caused very serious EV aggregation. So they could see very much bigger size of exosomes. And one doctor asked me the difference or similarity between exosomes or PDRN. So I tried to compare the exosomes against PDRN. So the number of this number is the total publication available on PubMed in November. So we found that the total number of publication on exosome is more than 35,000 publication, while the number of publication on PDRN is just 200. So the biggest difference is the ingredients because exosomes are supposed to deliver many kinds of ingredients at the same time. For example, last year there was another Nobel Prize in biology. So it's about microRNAs. So now microRNAs became Nobel Prize. I'll share more information later, but in our exosome, microRNA 7, LAT7 is one of the most important microRNA contained in our exosomes. And our exosome can deliver more than 1,000 growth factors, lipids, or microRNAs at the same time, which is much, much more potent than PDRN. Because PDRN is just one type of small DNA fragment. And you can see that in terms of the number of clinical studies, you can find 95 publication clinical studies registered on PubMed, while you can find only 17 publications on PubMed. And in terms of mechanisms, exosomes can give you very strong regeneration, skin barrier remodeling, and strong anti-inflammatory effects. So in any case, you can see that exosomes have much more potential. For example, last year in Dubai, and one third-party company compared the potential of different technology, for example, EGF, PDRN, PDLA, and exosome, and they found that in terms of wound regeneration, exosomes are the best, and also they found that the exosome treatment from this kind of animal study, they could see the biggest amount of the filaggrin protein, which is the key skin barrier protein, and the Ki67, which is the biomarker of cellular proliferation in our exosome treated group. So here again, our products, our ingredients can clearly show the extracellular vesicles or exosomes. And from this part, my third topic for today is about lipids, and then it's brand new scientific discoveries from our team, which is related to skin barrier reconstruction and biological functions. So, exosomes are surrounded by lipid membrane, and they are containing lipids as well as lipid generating enzymes. So we call them uh, tools and workers together existing in exosomes. And then we try to profile all the contents of lipids from this kind of analysis. And then we could see the very consistent composition of the major lipid components from phospholipids, sphingolipids, something like that. And our skin barrier is one of the most important part of our skin. And from our study of second publication, our exosomes can induce de novo synthesis of ceramide or dihydroceramides, which are very critical for healthy skin barrier. And also, our exosomes could reduce the amount of TSLP, which is a key itching molecule. So after that, for last 3 years, my team tried to profile or to make extensive lipidomic analysis, and just we submitted based on our efforts. So my team found very important 5 classes of more than 227 lipids identified, and we could see 16 ceramide metabolic enzymes. So this kind of combination of lipid or lipid generating enzyme can maintain the integrity of the membrane structure of exosomes, and they can deliver important biological functions to another cells. So, for example, our exosomes, adipose stem cell exosome, can increase the amount of S1P. S1P is a very important signaling molecule to stimulate keratinocyte differentiation, or to increase AMP production, or to promote wound healing. And as you can see here, this data will be published in a few months. Uh, so IL-4, we try to check the expression of the IL-4 from this in vitro study. So the key is that our exosomes, in combination with sphingosine, this combination can reduce the amount of IL-4. IL-4 is one of the major cytokines for dermatitis, something like that. And also, we found that the combination of our exosome and sphingosine was able to increase the production of filaggrin gene expression. So our products are containing very important enzymes to increase the cellular level of S1P, which can do a lot of good things for our skin. So this is a summary of our lipidomic analysis. And then from IMCAS, we will share more data. And this part is about exosomal microRNAs. And Dr. Lin shared some information on our microRNAs. And you can see here, microRNAs became another Nobel Prize last year. And the microRNAs, the main function of a microRNA is to block the translation, so it can block the protein expression. So the key is that these Nobel laureates cloned the first microRNA from human cells. It's microRNA L87. So we try to check the existence of L87 microRNA from our human adipose stem cell exosomes or Rose stem cell exosomes, and we found that both of our exosomes are containing high amount of micron L87. And we found that our human stem cell exosomes are containing LAT7B and 7A and or other different types of LT7 family. And the top 20 microRNAs are existing of how can I say, consist of 70% of the total microRNA expression. So scientists already made a lot of publication on the biological variety of biological functions for our skin. As you can see here, LT7 is very important for anti-inflammation, and micron 21 is very important for proliferation, and 29, 23 is very important for anti-scarring. So I will show you more and more. And here, the main biological function is of LT7B is anti-cancer or anti-inflammatory effects. And I told you that LT7 is the single most abundant microRNA in our human stem cell exosomes. And very fortunately, the canine mesenchymal stem cell exosomes, I mean canine adipose stem cell exosomes also have a very high amount of LAT7B and A, like human adipose stem cell exosomes. And here, the main function of microRNA 29 is about anti-keloid, and the main function of the microRNA is 141 and 200 is anti-melanogenesis. And again, here, microRNA 125B is anti-melanogenesis. Micron 21 is also. So here, when you compare the left panel and the right panel, we can show our stem cell exosomes are containing very important microRNAs for our skin. And Dr. Lin already mentioned the case that our rose stem cell exosomes are containing several important microRNAs for collagen synthesis, anti-scarring, or anti-inflammation. So here, this part is about proteins. So a few important proteins like these exosome biomarkers inside or outside or enzymes. So Dr. Carlos mentioned this, all these important molecules on the surface or inside of our exosome, and our team made extensive proteomic analysis to compare different batches of our human stem cell exosomes or our rose stem cell exosomes. And the key is that the proteins are usually responsible for cellular proliferation or wound healing, which is a regeneration. And we shared all the ingredients in our second publication. And this is a part of our data. So you can see a lot of nice growth factors contained in our exosome production batches. And one important enzyme is CD73 on the surface of exosome. They are very important. They can induce very strong paracrine immunosuppression by converting adenosine monophosphate into adenosine. Also, very recently, my team found that very important protein named 1433, and this is a very well-known protein for longevity. So I hope you can check this publication, or we can share this publication. And from our proteomic analysis of our large stem cell exosomes, we could see very new types of proteins, which are very different from those of humans, but they are very responsible for they have very different types of molecular functions, biological process, or cellular components. My team will do more research to identify very new or innovative types of mechanism of action. So here again, uh, my team, this is the summary. So my team isolated stem cells from roses, and when they culture, they released very different classes of exosomes. And when we treated rose stem cell exosomes, they can deliver important molecules for proliferation or to release more exosomes. And we have done all the characterization studies like this. You can see that this is the biomarker, exosome specific markers named CD63 on the surface of human stem cell exosome. And scientists found that a very similar exosome specific biomarker named TET8. So we found that our large stem cell exosome, this kind of very specific or exosome specific biomarker on the surface of our exosomes. And here, another dermatologist questioned me how large stem cell exosome can induce neocollagenesis. So I tried to find more references. So this is very important because microRNA 574 can induce neocollagenesis. So the data, I'll show you the data here. Scientists found that when they treated microRNA 574, they can induce more collagen. And another example is that beta vulgaris juice. They are containing exosome-like nanoparticles. And from their data, you can see that more type 1 collagen expression from the treatment of the vulgaris. And this is another example of wheat-derived nanovesicles can induce wound healing activities. And one of their data is that wheat exosome can induce more expression of type 1 collagen A. And this is another interesting example of sea plant named Eclonia cava, and the nanoparticles isolated from Eclonia cava can induce more collagen, or they can promote very nice wound healing from this animal study. And our low stem cell exosomes definitely are containing high amount of 547, but another collagen-inducing microRNA is the microRNA 1246. So scientists already found that microRNA 1246 can induce more procollagen type 1, as you can see here. And our large stem cell exosome contains another type of microRNA 130A. 130A has a very fiber proliferative potential. So when they treated microRNA 130A, they can see that more expression of the or more cellular proliferation. And our large stem cell exosomes are containing microRNA 122, and scientists already published that the adipose stem cell exosomes are containing microRNA 122. So this microRNA 122 is shared by human stem cell exosomes as well as our large stem cell exosome, and they can inhibit the function of dihydrotestosterone. So it's very surprising to see that this shared microRNA 122 can induce hair, or they can prevent hair loss. And again here, LT7B, they are very anti-inflammatory, but in another publication, we could see that LT7B can induce longer hair in alpaca by downregulating ectodysplacin A. So it's very nice science, but a little complicated. So in summary, when you look at here again, this is the profiling data, part of our profiling data of our raw stem cell exosome. LT7B, 122 for hair, 138 for cellular proliferation, 21, 23, 29 is for anti-scarring. 125 is anti-melanogenesis. 574 is neocollagenesis. 1246 is also cellular proliferation or neocollagenesis. And here you can see that we have ExoBio is the only exosome company who achieved this kind of patent on plant stem cell exosomes. Again, we registered our raw stem cell exosome as a cosmetic ingredients.